Hello, and welcome to our video on antibody-mediated immune response. Immunity is the ability of the body to resist any infection or resistance against the growth of foreign bodies in the body. The immune system is the self-defense mechanism developed by our body to protect itself from foreign invasion of harmful microbes or different types of infections. The human body consists of several specialized cells that make up the entire immune system, with each cell serving its own function. These are known as the white blood cells, WBCs, and can be termed as the soldiers of the body. WBCs are highly differentiated, and there are several types to go through. Lymphocytes are the agranulated WBCs primarily involved in the immune responses of our body. They originate from bone marrow, and they're found in the body fluids, blood, and lymph. Different types of lymphocytes involved in immune responses are T cells, or T lymphocytes. T stands for thymus, the organ in which these cells mature. T cells are involved in cell-mediated immunity. B cells, also called B lymphocytes. Here, the B stands for bone marrow, and hence they mature in the bone marrow. B cells are primarily responsible for humoral immunity. B cells, along with T cells, are major components of the adaptive immune response system. Natural killer cells, also known as NK cells, a type of cytotoxic lymphocyte, and they're primarily responsible for the innate immune system. Other types of cells that are part of the immune system include the following: neutrophils, granulated white blood cells that are responsible for the inflammatory action that helps the body in preparing a proper immune response. They make up 60 to 65% of total white blood cells. Monocytes, agranular white blood cells making up 6 to 8% of the total white blood cell count, and are among the largest ones. These are phagocytic in nature, and they have the ability to destroy invaders. Monocytes are formed in the bone marrow when transferred to blood, where they circulate for 2 to 3 days before entering tissue, where they become macrophages or dendritic cells, antigen-presenting cells. Eosinophils are granulated white blood cells that promote inflammation. These cells are active in an allergic reaction. They modulate inflammatory responses and trap and kill cells infected with bacteria or parasites. Basophils are granulated white blood cells that are in the least amount in the body. They make up less than 1% of the total count. They are responsible for allergic reactions, as they have histamine in them. The first line of defense against non-self pathogens is the innate or nonspecific immune response. The innate immune response consists of physical, chemical, and cellular defenses against pathogens. The main purpose of the innate immune system is to immediately prevent the spread and movement of foreign pathogens throughout the body. The second line of defense against non-self pathogens is called adaptive immune response. Adaptive immunity is also referred to as acquired immunity or specific immunity, and it's only found in vertebrates. The adaptive immune response is specific to the pathogen presented. The adaptive immune response is meant to attack non-self pathogens, but can sometimes make errors and attack itself. When this happens, autoimmune disease can develop, for example, lupus or rheumatoid arthritis. The hallmark of the adaptive immune system is clonal expansion of lymphocytes. Clonal expansion is the rapid increase of T and B lymphocytes from one or a few cells to millions. Each clone that originates from the original T or B lymphocyte has the same antigen receptor as the original and fights the same pathogen. While the innate immune response is immediate, the adaptive immune response is not. However, the effect of the adaptive immune response is long-lasting, highly specific, and is sustained long-term by memory T cells. There are two main mechanisms of immunity within the adaptive immune system: humoral and cellular. Humoral immunity is also called antibody-mediated immunity. With the assistance from helper T cells, B cells will differentiate into plasma B cells that can produce antibodies against a specific antigen. The humoral immune system deals with antigens from pathogens that are freely circulating or outside the infected cells. Antibodies produced by the B cells will bind to antigens, neutralizing them or causing lysis, dissolution or destruction of cells by a lysin, or phagocytosis. Cellular immunity occurs inside infected cells and is mediated by T lymphocytes. The pathogen's antigens are expressed on the cell's surface, or on an antigen-presenting cell. Helper T cells release cytokines that help activated T cells bind to the infected cell's MHC antigen complex and differentiate the T cell into a cytotoxic T cell. The infected cell then undergoes lysis. Humoral versus cell-mediated. B cells activate humoral immunity, whereas T cells activate cell-mediated immunity. The major difference between humoral and cell-mediated immunity is that humoral immunity produces antigen-specific antibodies, whereas cell-mediated immunity does not. T lymphocytes, on the other hand, kill infected cells by triggering apoptosis. Humoral immunity develops quickly, whereas cell-mediated immunity takes considerably longer. Extracellular microorganisms and their poisons are targeted by humoral immunity. Intracellular microorganisms, such as bacteria and tumor cells, are targets of cell-mediated immunity. The LG, CD40, CD21, and FC receptors are the humoral immunity's accessory receptors. The accessory receptors of cell-mediated immunity are CD2, CD3, CD4, CD8, CD28, and integrins. Humoral immunity recognizes the unprocessed antigens. In humoral immunity, plasma B cells release antibodies. Cytokines are released by T cells. Tumor cells and transplants are immune to humoral immunity. Tumor cells and transplants are both affected by cell-mediated immunity. Humoral immunity is extremely important in both health and disease, and it can be both harmful and useful. Antibody-mediated protection against pathogens induced by vaccines or infections is crucial in host defense, but pathogen-specific antibodies can also promote infectious processes or drive pathology. Loss of immunological tolerance is linked to the generation of self-reactive antibodies, which can aggravate the condition, and loss of growth control can lead to a variety of B-cell malignancies. When foreign material, antigens, are recognized in the body responds with an antibody-mediated reaction. Extracellular intruders, such as bacteria, are commonly found in this foreign material. B-cell lymphocytes, a type of immune cell that makes antibodies after detecting a specific antigen, are principally responsible for this method. Lymphocytes known as naive B cells circulate throughout the body via the lymphatic system. These cells produce antigen-specific molecules that are necessary for detecting infectious pathogens in the human body. When naive B cells in the lymphatic system come into contact with an antigen, they begin the differentiation process that results in the formation of memory B cells and effector B cells. Memory B cells and effector B cells produce the same antigen-specific molecules as their parent naive B cell during this development. The activated memory B cells express these antigen-specific molecules on their surface with the help of T cell lymphocytes, which are activated by MHC class II receptors that recognize microbial-associated antigens. The effector B cells secrete these molecules in the blood to bind the antigen of interest. Humoral immunity is the aspect of immunity that is mediated by macromolecules, including secreted antibodies, complement proteins, and certain antimicrobial peptides located in extracellular fluids. Humoral immunity is named so because it involves substances found in the humors, or body fluids. It contrasts with cell-mediated immunity. Humoral immunity refers to antibody production and the coinciding processes that accompany it, including the following: Th2 activation and cytokine production, germinal center formation and isotope switching, and affinity maturation and memory cell generation. It also refers to the effector functions of antibodies, which include pathogen and toxin neutralization, classical complement activation, and opsonin promotion of phagocytosis, and pathogen elimination. 1. Antigen recognition. B cells have specific receptors on their surface that can recognize and bind to unique molecules on the surface of foreign invaders, called antigens. Antigens are typically proteins or other large molecules found on the surface of pathogens. 2. Activation and clonal expansion. When a B cell encounters an antigen that matches its receptor, it becomes activated. This activation triggers the B cell to undergo clonal expansion, producing numerous identical copies of itself called plasma cells. 3. Antibody production. Plasma cells are specialized B cells that secrete large quantities of antibodies into the bloodstream and tissues. Antibodies are Y-shaped proteins that have binding sites specifically designed to attach to the antigens of the invading pathogen. 4. Neutralization. Antibodies bind to the antigens on the surface of the pathogen, effectively neutralizing it. This binding can prevent the pathogen from infecting host cells and also marks it for destruction by other components of the immune system. 5. Opsonization. Antibodies can act as opsonins, substances that coat the pathogen, making it more recognizable to phagocytic cells, for example macrophages, that engulf and destroy the invader. 6. Complement activation. Antibodies can trigger the activation of the complement system, a group of proteins that cause cell lysis, inflammation, and the recruitment of immune cells to the site of infection. 7. Memory B cells. During the immune response, some B cells differentiate into memory B cells. These cells remember the encountered antigen, allowing the immune system to mount a faster and more robust response if the same pathogen is encountered again in the future. Antibodies are heavy proteins that are approximately 10 nanometers in size. These molecules are produced by B cells in order to identify and neutralize harmful agents such as infectious bacteria, fungi, and viruses. These Y-shaped proteins contain antigen-binding sites that specifically bind to their target antigens. Once antibodies effectively bind to their target antigen, they can either neutralize their target antigen directly by blocking normal antigen binding, or they can induce the recruitment of other immune cells or molecules that promote the antigen's removal or destruction. In mammals, such antibodies come in a variety of forms, commonly known as isotypes. An incompatible blood transfusion causes a transfusion reaction, which is mediated by the humoral immune response. This type of reaction, called an acute hemolytic reaction, results in the rapid destruction, hemolysis, of the donor red blood cells by host antibodies. The cause is usually a clerical error, such as the wrong unit of blood being given to the wrong patient. The symptoms are fever and chills, sometimes with back pain, and pink or red urine, hemoglobinuria. The major complication is that hemoglobin released by the destruction of red blood cells can cause acute kidney failure. Each blood cell produces its own set of antibodies with unique antigen-specific bonding sites. Initially, naive B cells produce antibodies that remain bound to the cellular surface so that their exposed antigen binding sites can detect potential pathogens, toxins, and foreign material. This surface-bound form of an antibody is known as immunoglobulin. When an antigen matching the antigen binding site binds to a naive or memory B cell, it activates the B cell to produce and secrete more antigen-specific antibodies. Once a B cell fully matures, it's known as a plasma cell and will continue to produce and secrete antigen-specific antibodies for the remainder of its life cycle. Classes of immunoglobulins, antibodies. IgG, the most abundant antibody, found in blood, lymph, and intestines. It protects against viruses and bacteria. IgA, found mainly in sweat, tears, saliva, mucus, milk, and GI secretions. It provides localized protection on mucous membranes against bacteria and viruses. IgM, the first antibody class to be secreted by plasma cells after initial exposure to an antigen. It's found in the blood, lymph, and as antigen receptors on surface of B cells. IgD, found in blood, lymph, and as antigen receptors on the surface of B cells. It's involved in the activation of B cells. IgE, the least abundant of all antibodies, found on mast cells and basophils, involved in allergic and hypersensitivity reactions, and provides protection against parasitic worms. The complement system is a biochemical cascade of the innate immune system that helps clear pathogens from an organism. It's derived from many small blood plasma proteins that work together to disrupt the target cell's plasma membrane, leading to cytolysis of the cell. The complement system consists of more than 35 soluble and cell-bound proteins, 12 of which are directly involved in the complement pathways. The complement system is involved in the activities of both innate immunity and acquired immunity. Activation of this system leads to cytolysis, chemotaxis, opsonization, immune clearance, and inflammation, as well as the marking of pathogens for phagocytosis. The proteins account for 5% of the serum globulin fraction. Most of these proteins circulate as zymogens, which are inactive until proteolytic cleavage. 3 biochemical pathways activate the complement system: the classical complement pathway, the alternate complement pathway, and the mannose-binding lectin pathway. The classical complement pathway typically requires antibodies for activation and is a specific immune response, while the alternate pathway can be activated without the presence of antibodies and is considered a non-specific immune response. B-cell activation is a large part of the humoral immune response. These processes differ only in the process of activating C3 convertase, which is the initial step of complement activation, and the subsequent process are eventually the same, forming a structure called the membrane attack complex, the MAC, on the bacterial cell wall to destroy the bacteria. Thank you for watching, and have a great day.